I think, given what Catherine in particular presented this morning, I think the title is slightly misleading because it's not just methodological issues, it's also, I think, some technical issues and some practical issues as well. So we've kind of bunched them all together. So it's not just methodological issues. This is going to be a team effort, so it's not just myself, it's also Gilles who's sat at the back there. We've both been working at HERC for some time, myself for about 10 years, Gilles for a couple of years now, on economic evaluations in genetics and in genomics, and hopefully we can provide a little bit of insight today into the sort of challenges that we face when we actually do these economic evaluations. So I'll go through the literature first of all, and then Gilles will hopefully give you a nice example at the end which will illustrate some of these challenges. I'm not sure how much of this is going to be how, but hopefully some of it is going to be how. So what I'm going to talk about is a literature review that Sarah and I published probably about 18 months ago now in Pharmacogenomics. I'll draw on this over the next few slides, and it was mentioned, I think, by Christine earlier on. What I'm not going to do is dwell too much on the methods, but I will say a couple of things. Firstly, that the aims that we had when we did this review were to pick up references that looked at challenges in this area when we're doing economic evaluations, and also to try and establish, is genomics exceptional? Is there a consensus as to whether genomics is a special case in terms of health economics? So we picked up about 50 references. Most of these were fairly recent, as you'd expect, and the issues fell into 4 categories, plus a sort of miscellaneous category as well. So the first group of challenges related to the analytical approach. And I think there were 3 things that we'd want to pick out here. There's going to be some overlap here with what people said earlier, and I'll try and sort of speed through those bits. The first challenge relates to the perspective that you might want to adopt when you're doing an economic evaluation in genomics. So generally speaking, as health economists, we're sort of conditioned to almost always take a healthcare perspective, but in genomics you're often faced with test results that have an impact on not just healthcare but on life decisions as well. So there's a suggestion that maybe we should be thinking about a societal perspective more often in genomics. We also need to think quite carefully about when we time economic evaluations. So Gilles will come back to this point later, and I suspect Martin, wherever he is, will possibly touch on this in his session next. As we all know, test attributes change quite quickly over time. Tests evolve. For newer tests in particular, information on test attributes is often lacking. New patient subgroups are constantly discovered, and each of these has different clinical implications. So the question for us, I guess, as health economists is, given this, how can we best use limited health economic evaluation resources to, when the science moves so quickly? And then the analytical context as well is crucial. So tests often have multiple applications in a variety of different disease contexts, and these different applications might not always be similarly cost effective. So a good example of this is the use of Oncotype DX. It's differently cost effective in breast cancer and colon cancer. Choice of comparator is also important. As most probably realise, standard genomic testing practice rarely exists, so you have to have a think at the start of your economic evaluation about what exactly your comparator is going to be. And we also need to make sure that we include all subsequent therapeutic decisions and the costs associated with these when we do these sort of economic evaluations. Now, the case study that Gilles is going to discuss in a minute will touch on this point, and I've certainly found this in work that I've been doing in targeted sequencing and chronic lymphocytic leukemia as well. So that's the analytical approach. There are also a whole bunch of issues surrounding costs and resource use. And the first point I guess to note is that there's a broad range of costs that you need to think about when you're doing economic evaluations in this context. Now, to a certain extent, this isn't any different to when you're doing economic evaluations of non-genomic interventions, but it does mean that you have to quite carefully plan data collection in genomics. Now, I'm not going to go through this whole list of costs, but I will mention a couple that seem to me to be particularly important, and the first of these is indirect cost to patients at the bottom of this list. Obviously, whether we include these or not depends on the analytical perspective that we adopt, which links back to the earlier slide. But there is evidence, for example, going back to Oncotype DX again, that it becomes incredibly cost effective when you start including the productivity costs associated with the fact that people don't need to take as much time off work now due to the effects of unnecessary treatment. And then the other group of costs I wanted to mention is the last group on the list, which is infrastructure costs. So the problem that we have often when we're thinking about sequencing is that genomic platform diagnostics often have multiple applications. So you may install a piece of equipment for one use only, but once it's in place there are other test applications that suddenly become an option. So how do we allocate these infrastructure costs? If we overburden initial applications with too many of these infrastructure costs compared to later applications, then any economic evaluations aren't probably going to paint a true picture of the value associated with these different applications. Second category of issues relates to how much genomic tests cost. Most of the time we don't have a great idea of how much genomic tests cost, and we have touched on this in previous sessions. There are no national guidelines, there are no sort of nationally agreed reimbursement rates for genomic tests. If you need a cost for a genomic test, often the only option is to go out and cost it yourself. The problem you have here is that test costs obviously vary from one lab to the next, and you have issues regarding the generalisability of these costs. And it's a problem because HTA agencies such as NICE require that nationally agreed prices are included in their economic evaluations. And then finally, you need to think about when to collect cost data. We have a couple of problems here. So firstly, tumours evolve over time, tumours acquire new mutations. These all have different clinical implications that you might want to take into account, so you might need to repeat genetic tests over time. Obviously this has a cost implication. And similarly, the literature doesn't stand still, it moves on, and tests are often packaged with filters that present a sort of subset of information to clinicians, just the information they need to make a decision. As the literature evolves, the amount of information that could be presented to clinicians changes, so you may need to update the filters, and then you need to think about retesting patient samples, and again this has cost implications. Okay, measuring outcomes. I think for me at least this is possibly where the most interesting challenges lie, certainly probably the challenges that provoke the most debate in this area. And the first problem that we have is that commonly used outcome measures don't really do a good job in terms of evaluating genomic interventions. They're not particularly good at capturing the impact of possessing additional diagnostic information, for example. Often we don't have information on the typical health states that patients are in following testing, so for example living with the knowledge that you have a mutation that may have implications in the future. So consequently, if the outcomes measure that you're using in your economic evaluation is unlikely to show anything other than a negligible difference between current practice and potential future practice, what you're going to end up with is quite high ICEs, which are obviously going to impact on adoption decisions. So these other impacts that genomic tests can have are often captured using the umbrella term personal utility, which we've touched on in previous sessions. This is the benefits or harms that are manifested outside of medical contexts, and they're a whole bunch of different positive and negative effects. So for example, improvements in understanding, giving a sense of control to patients, and also sort of negative effects such as increasing anxiety. So there are some potentially important impacts associated with genomic testing that we don't, as a rule, include because standard metrics miss these benefits and these positive and negative effects. So that leads me on to cost-benefit analysis. Given that it could potentially be beneficial to include information on personal utility in economic evaluations in genomics, cost-benefit analysis, or the welfare risk perspective, may have a greater role to play. Obviously, there are several different ways to monetize health outcomes in a cost-benefit analysis. We tended to focus so far in Oxford on DCEs, but that's not to say that these other approaches aren't also relevant, and in fact in some cases they may be potentially more useful than DCEs. But the problem we have when applying the cost-benefit analysis approach is that most, if not all, HTA agencies favour using cost-utility analysis using QALYs. There are a few agencies that sometimes permit us to submit supplementary analyses, which could take the form of cost-benefit analysis. I think Australia and Canada are two examples, and perhaps public health interventions in the UK as well. But generally speaking, cost-benefit analyses, which may do a better job of evaluating these interventions, they don't tend to influence decision making in healthcare at the moment. Okay, the last category of issues relates to the measurement of effectiveness, and again I think there's three things to think about here. The first of these relates to patient and clinician behaviour in this context. So we don't have a great deal of information on how patients and clinicians will use these tests. how they will respond to test results. But this information should be feeding into economic evaluations. There's a little bit of limited evidence from the US that if you don't mandate testing, then uptake isn't universal, and if this is the case, then as I say, this should be feeding into economic evaluations. I think we're all probably familiar with the idea that the sort of data that we're working with, the sort of effectiveness data that we have access to in genomics is often weak. It's difficult to conduct randomised controlled trials for many of these interventions. There are various alternatives. I won't talk about the advantages and disadvantages of each, but obviously there are some obvious advantages and disadvantages for each. One possibility in genomics and health economics is that maybe we want to start thinking about generating more practice-based evidence in post-implementation studies, but that's not without difficulties as well, because obviously you have issues around disinvestment when tests are already in place. And then the final thing I wanted to mention in relation to effectiveness is that the data that we're working with in these economic evaluations tends to be very complex. And there's quite a limited evidence base linking genomic data with health outcomes. One of the key problems we've got here is that testing outcomes in genomics are influenced by multiple genes. Each mutation can impact on multiple outcomes, and the influence of mutation on a given outcome can vary from one individual to the next. Different solutions have been suggested in the literature. One option is the use of polygenic risk score, so somehow combining all of this information into one measure. This needs a lot of data. It's difficult to reproduce across different studies, and decision makers may find it difficult to synthesise this sort of evidence, but it's one possible approach going forward. There are a few issues that fell outside of these four categories. I'm not going to look at all of them, but one in particular I wanted to mention, which we haven't really touched on today, is research prioritisation. Several authors have noted that value of information analysis could be important in genomics. We've already said there's weak effectiveness data for many genomic interventions, so additional research in this area is obviously going to be welcome, but if we don't have options such as randomised controlled trials, which is often the case, the benefits of this future research need to be compared quite carefully with the costs before we allocate scarce research funding in one direction or another. And there aren't many value of information analyses done in this area at the moment, and I suggest that may need to change if we want to have a more influence on decision making in this context. Okay, final slide from me, and then I'll hand over to Gilles. That's a really brief run through of the issues that we found in the literature. I'd encourage you to go to the paper if you want any more information. There are obviously a couple of different perspectives you can take as to whether genomics is exceptional, whether it poses an exceptional challenge in this area. I put a couple of quotes up here that perhaps illustrate different perspectives on this. Wolf is in the room if you want to disagree with his comment at the bottom. I guess where I currently stand on this is that the quantity and range of challenges in genomics means that economic evaluations of these technologies pose a particular challenge for us as health economists, and it's not so much that the uniqueness doesn't relate to each challenge individually, but more to the collection of all of these challenges for interventions in genomics. But as I say, there are as many opinions on this as there are people in the room, so you may all think differently on that. I'm going to stop here. I'm going to hand over to Gilles, and what Gilles is going to do is present a case study which hopefully quite nicely illustrates some of these challenges.
So in this part of the presentation, I'll be finding some practical issues that should complementary the examples that James provided. So these examples are taken from from a study that was funded by the Technology Strategy Board, and this was a So this was a study on tumor profiling to improve cancer care, and in 2011 there was a funding application on this tumor profiling, and one of the requirements that was set by Cancer Research UK was that the cost of a cancer panel should cost £300 or less. And as James already mentioned, there's no national guidelines about how much a genomic test should cost. So I was interested in what this number is based on, and in terms of cost breakdown and what it would cover. So I contacted Cancer Research UK and asked them about this. And actually, no one could provide a satisfactory answer. This might have been to internal changes in terms of people moving positions. But the answer that I did get was that this is the cost that Cancer Research UK was willing to pay and that should be achieved relating to current clinical practice. And this cost would cover the cancer panel itself, as well tissue preparation and analysis. So I won't be discussing the study in itself, but I'm just picking out a few examples. So this study was done in collaboration with the Oxford Molecular Diagnostic Centre. So it was within an NHS setting. And the main objective for the health economics component was to estimate costs using a multi-gene cancer panel. And this was the ion-torrent personal genome machine of Life Technologies versus a single gene testing for cancer diagnosis and treatment. And this single gene platform was the Cobas Z480 by Roche. Based upon standard operating procedures, we developed cost questionnaires. There are two versions, one for PGM and one for Cobas. And these were there to cover the resource use. And resource items that were included were staff time, consumables, equipment, error rates, etc. Once we had this cost, we costed several scenarios. And this was the PGM in itself, if you're using a clinical pathway, but as well combining these with different tests. For example, first having a patient tested by the EGFR and Cobas, followed by PGM, or depending on the clinical context, combining EGFR tests, BRAF and KRAS. So we looked particularly in the concept of lung cancer, and we conducted cost effectiveness analysis as well, and we looked as well at some of bowel. The multi-gene panel, so the PGM, was estimated to cost £339, which is actually quite close to the estimate that Cancer Research UK set. And for single gene testing, this was between £71 and £140, and it depends on the mutation testing that you want to do. So the first example is the rate of technological advancement. In genomics, it's going at such a pace that you can try to prepare as much as possible. However, what we had is once we finalized the cost questionnaires, and I think 1 or 2 weeks after, some of the steps were already automatized, so reducing hands-on time. So we had to make amendments. Also, once we had the cost data, we were doing the analysis. Some of the steps further were automatized by, for example, the Iron Chef. So the Iron Chef would reduce hands-on time about 50 minutes in terms of chip loading. And for the Oxford lab, this would increase the cost per sample about £5. However, if they would increase the weekly throughput by one sample, this would be already accounted for. Then there's another machine. James already mentioned this. Is that if new technology becomes available, this might actually change the cost and change the whole analysis that you've conducted. So this Kia Symphony can automatize the DNA scraping procedure, which also reduces hands-on time. Initially, this would be reducing cost per sample by £2 per sample, which is different from the Iron Chef. The Iron Chef is very expensive in itself. However, the cost for the Kia Symphony is covered by the mutation kits, or the DNA kit, sorry. There's also a robot available that could automatize further activities, could reduce error rates, and that could change the cost you've estimated as well. In addition, there were 2 new mutation kits that became available, which was the NRAS and PIK3CA. These were by Roche, and NRAS testing was costed at £87 per sample, and PIK3CA £239 per sample. And these were accounted for in the sensitivity analysis. So the second example is on testing guidelines. James already mentioned that the choice of comparator is important. However, standard genomic testing practice does not exist. So if you look at the guidelines of quality assessment, they vary per country. But for the UK, if you would test in the context of lung cancer, you want to test for EGFR, KRAS, BRAF, and PIK3CA. For bowel cancer, it's KRAS, BRAF, PIK3CA, and NRAS. However, they do not match clinical or current clinical practice. So in the Oxford lab, if someone is being diagnosed with lung cancer, they would initially test for EGFR, so the single gene test on Cobas, followed by PGM, which would cover the additional genes as well. For bowel cancer, they would initially just test on PGM, because for NRAS the shelf life is quite short. But there's a difference in terms of the clinical practice and guidelines. If you test on the PGM, that would be £339. For lung cancer, if you test on the COBAS, that would be £495, and for bowel cancer, that would be £477. In the analysis, we made a combination of both the clinical practice strategies and as well those in the quality guidelines. So the last example is clinical context, because the platform in itself can be cost effective, but it does depend on the clinical context. And this has been mentioned in some presentations earlier today. Also the ICEs, the incremental cost effectiveness ratios, can change instantly depending on your clinical context. For example, lung cancer, the ICE was estimated to be £1,689, which is about €2,200 or $2,600 per life year gained. So to clarify, in the decision model, we looked at multiple strategies, and we included not just the platform, but as well treatment, and it was best standard care, including chemo, or best standard care and targeted. And we looked at lung cancer mainly, and as well in bowel cancer. And in terms of effectiveness data, we looked at life years gained, both progression-free survival and overall survival. And then we looked as well at quality data. And James already mentioned actually that effectiveness data is quite limited, so that made it quite challenging as well. So in lung cancer, so initially if you were looking at the life years gained, there would be an ISO of £689, and this was measured in progression-free survival. But not all studies have the same definition of progression-free survival, so it's quite an ambiguous term. So we looked at overall survival as well, and targeted treatment did substantially better, so there's a bigger change which resulted in a lower ISO of £425, which is €585 or $655 per life years gained. When we're using quality data, actually none of the genetic or genomic platforms were recommended because there was no difference in the literature between the two. For bowel cancer, the ISO for the PGM was actually quite high. It was about £98,681, which is about €135,000 or $152,000. However, as James already mentioned, that the quality data, the information that's available on effectiveness is quite limited. What we did as well is trying to look to see if we could change these outcomes by separating between first line and second line treatment. However, there's just too large a variation between the different studies, which made the analysis quite challenging. One of the reasons why the ISO for the PGM in the context of bowel cancer is quite high as well is because of the treatment. So if you look at the treatment for bowel cancer, this is about £20,000, 687 for targeted treatment versus £2,300 for best standard care. So the effectiveness difference was actually quite small, but the cost difference was extremely large. There's obviously various other strategies that you come up with. However, we assumed that the health outcomes was the same for patients with or without mutation when tested positive. However, there's also some evidence suggesting that those with wild-type EGFR mutation receive targeted treatment would actually be benefiting more from chemotherapy than from targeted treatment, or might even do worse. So this will change your outcome as well. So in summary, cost effectiveness for genomic testing really depends on the context in which you place it, and as well what kind of treatment is being given. So hopefully these examples on the terms of the rate of technological advancement, the mismatch of testing guidelines and clinical practice, and the influence of clinical context on your cost effectiveness have complemented the examples that James provided. In particular, we would like to thank our supervisor, Dr. Sarah Wordsworth, and Dr. Anna Shu from the Oxford Molecular Diagnostics Center, our funders, and all the others that were involved in these studies. Thank you.